All right, it's 12:30, so let's go ahead and get started. Welcome. Thank you for joining the N of One Collaborative Seminar Series. My name is Nicole Nolan. I am N1C's Programs Manager. I'm going to kick us off with just a few housekeeping announcements, and then I'll pass it over to our moderator for today, Dr. Tim Yu. We'd like to thank our partner, the Chan Zuckerberg Initiative, for their support of N1C's mission. I'm going to drop my email in the chat, and I would love to hear from you if you or your organization is interested in learning more about how you can support the N1C. Additionally, an exciting announcement for today is that we just launched registration for our 2026 annual meeting, which will be held this year in Denver, Colorado on October 10th. For any of you attending the OTS annual meeting, that is one day prior in the same location. So we hope that you'll be able to adjust your travel and join us one day early. I will also be putting a link to register in the chat, and we hope to see you there. Lastly, I wanted to let you know that conversation and follow-up discussion from today's seminar will continue on Connections, N1C's online platform built to support collaboration virtually. So please join us if you have any thoughts after this seminar. We'd love to hear from you. And with that, I will turn it over to Dr. Tim Yu, N1C's co-founder, who has volunteered to moderate today's talk. Tim?
Thanks, Nicole, and hello, everybody. It's wonderful to have you here today. First, a couple of logistical reminders. We do encourage folks to turn on your cameras if you're able. We like to promote this session as an interactive session, a real chance to interact with a real-world leader that we have the pleasure of hosting today, Anastasia Kvarvarova. If you have any questions, however, please don't interrupt in the middle, but put them in the chat for discussion after the presentation. And then, but with your cameras on, we'll be able to get you to actually present the questions live, face-to-face, after Anastasia is done. As Nicole mentioned, our seminars with speakers' permission are recorded, so you can view them later on Connections. And Anastasia has graciously granted us permission to record her talk, so you'll be able to view it again afterwards. And just a post-seminar reminder, you will automatically receive a survey at the end of this that will pop up when you close the Zoom link. If you have just 30 seconds, we would hugely appreciate your feedback so we can continue to improve the session and host topics that continue to be interesting and valuable for all of you. With those logistics out of the way, it's with great personal and professional pleasure that I get to introduce Anastasia today. Anastasia is a professor at the University of Massachusetts Chan Medical School. She is also one of the primary faculty in the RNA Therapeutics Institute and the Program in Molecular Medicine, and the Raimondi Family Chair in Biomedical Research. Anastasia is a principal investigator on a host of fascinating projects to advance RNA therapeutics to the clinic for a broad range of medical indications with critical unmet needs. She has been one of the most influential folks to me personally in thinking about this space. With over 3 decades in advancing RNA therapies via both industry and academia. She's now authored more than 150 patents. She's a member of the National Academy of Inventors. She's co-founded several companies that are advancing RNA therapeutics in the clinic. And she's also a longstanding director of the Oligotherapeutics Society and a supporter of the N of One Collaborative for many years. To round off the embarrassing introductions, I just want to congratulate Anastasia for last year being awarded the Elsie Kroner Fresenius Prize for Medical Research. This is an international honor recognizing astonishing breakthroughs in biomedical research. She's only one of only 5 recipients of this honor since it was started 12 years ago. And I think hearing her talk, you'll understand why. Anastasia, your talk you are presenting today, her title is Developing Oligonucleotide Therapeutics for Rare Indications, an Academic Perspective. I'll pass it on to you.
Can you see the main screen? Yes.
Great.
Fantastic. Team, I cannot say no to you, so it's great to be here and share our perspective for what is going on in the venture of ever expanding development of oligonucleotide therapeutics. So if you look, and what I will be focusing today, it's on sRNA specifically. So if you look at the field of sRNA, it follows this absolutely typical hype curve. What it means that after fundamental biology was discovered, Craig Miller and Andrew Fire got the Nobel Prize, every single big pharma move in. There's tons of excitement. Things are being moved in the clinic, which are underdeveloped, and vast majority of them, actually all of them, do fail miserably, either through lack of efficacy or toxicity or combination of the above. Then chemists stay active, and 2010 was a very depressive time for the field, but they stay active. And as a result of this ongoing chemistry activity, there is now a vast portfolio of therapeutic platform which gives you an ability to develop drugs. And there is 8 approved, all of them in the liver. But what I will talk today is mostly about expanding this technology platform to deliver to tissues other than liver. And I think I will talk about 2 tissues, muscle and CNS, because both of them are very relevant and very commonly affected in N of 1 or ultra rare indications. So the beautiful thing about sRNA therapeutics that they work by a uniform mechanism. No matter how you deliver the oligos or sRNAs inside the cells, they are entrapped in endosomal lysosomal compartment, which serves as a depot of the drug. And then this depot of the drug is slowly releasing oligos over very long periods of time. Then it's loaded into the RIS complex, finds the target mRNA, cleaves, preventing its, a mutant protein or disease-causing protein from being expressed. And I think the fundamental advancement of this technology platform is the durability of the effect is extremely long. So normal right now in liver is 25 milligram quarterly or 200 milligram yearly. And this is not a mistake. We're talking about single administration of the drug gives you up to a year clinical duration of effect. And this is due to this predominantly factor of entrapment inside the endosomal lysosomal compartment and enzymatic mechanism of action where a very small number of loaded RIS complexes is sufficient to support robust silencing for extended periods of time. So because of this entrapment in endosomes and lysosomes has been so critical for durability of the effect. And I think the durability of effect, it really makes it specifically relevant for N-of-1 community because everything is associated with a lot of logistical as well as a poorly financial cost. And if you can reduce the number of administration of the drug to the patient, ideally to get to this point of every 6 months or every year, everything becomes much more feasible. So to be able to work for a year, it's absolutely essential to chemically stabilize or extensively chemically stabilize the sRNA cargo because endosomal lysosomal compartment is very aggressive in nucleases, and as a result of it, pretty much every single base and every single backbone need to be modified. So what you look over here, it's a pretty generic chemical architecture, and there are many of them right now, which combine a wide range of chemical modifications. So what is absolutely essential and non-negotiable? Every single base or every single sugar need to be modified with 2' methyls and 2' fluorous. There is a couple of positions in a guide strand, and the guide strand is a strand which will be entering into the Reese complex where it's critical to keep fluor because methyl or methoxyethyl modification, which is slightly more bulkier, yeah, in nature, it just doesn't fit. The most critical position is number 2 and number 14, but in generally, I will say 12 and 16, it's another positions we would like to keep as a fluora. In general, the more methyl modifications you have, the better, because it's going to be enhance the long-term stability of the duplex in this environment. But on top of it, it's critical to identify and optimize those chemical modification pattern in the way which would not interfere with efficiency of risk entry and risk assembly. Because if you do, you will lose in vivo potency. So in terms of sugar modifications, it's critical to have terminal stabilizing modification. And what is shown over here, that the 4 terminal backbones in red are modified with phosphatides. And phosphatides is a modification where non-bridging oxygen is substitute from O to S, and this is absolutely critical and non-negotiable for terminal stabilization and resistance against exonucleases. In some of the alnylam and GalNAc configurations, you see 3' end locking those phosphatide modifications because it's protected by the GalNAc. My recommendation, and I think most of the more modern advanced links, it's actually keep it. What is also important about the phosphatides is that while it's critical for stabilization, it's also enabling a lot of non-specific binding activity to other protein. In CNS in particular, the higher phosphatide content is associated with a higher toxicity, which is a very well-known and understood fact. For example, antisense compounds, which is another class of drugs, if it's fully phosphadilated, it's extremely toxic in the context of gapmers. So most of the current gapmers moving in the clinic have mixed backbone content where a reasonable number of phosphatides is substituted with phosphodiester. So Kenya Yamada Lab, which we work with a lot, recently has identified a novel type of stabilizing backbone, which called XNA, where the extra carbon is added to the backbone without affecting charge. And this modification is orthogonal to phosphatide, so it can be either used by itself or with phosphatides. And this is one of the current strategy how reduction in phosphatide content can be achieved without compromising the overall stabilization. And last and not least, it's E-vinyl phosphonate. It's modifying the phosphate of the 5' end of the guide, where the bridging now oxygen is substituted for the carbon and log at the chemical architectural configuration, which does favor interaction of the mid-domain of the EGA2. On top of it, methoxyethyl, which is slightly different, more bulky sugar modification, it's preferred to look at using certain position at the ends because it adds you more chemical stabilization. I understand that I go for a very long time kind of explaining the chemistry, but it really matters. It's all public information. We published this review in Nature Review Drug Discovery, look at the chemical configurations of the chemical configuration of some of the approved drugs, but the chemistry matter, and it's right now combination of multiple chemical together, which gives you this strong efficacy wide therapeutic index. So while the chemical architecture of the sRNA itself, it's independent of what type of delivery vehicle you're using, and most of the approved drugs in liver, they're using this GalNAc to enable delivery, and that is enabling delivery to hepatocytes and hepatocytes only. There's also a rising class with several compounds in clinics of antibody siRNA conjugates. That is a little bit more challenging for N-of-1 applications at this point because of the complexity of the CMC, and the tox profile is not very well understood, but this is coming of age and it's expanding in with a speed which is unprecedented. So I will say within very short period of time, we will have a collection of antibodies which are clinically validated and give you a robust delivery of the cargo, at least to heart and the muscle. And another way to deliver oligonucleotides, which are in clinic, is by changing their biophysical properties. So if you think about the traditional antisense in CNS, the delivery is not happening through the conjugation. Delivery is happening through presence of this phosphatide backbone, which enable binding to different proteins. And the way we can manipulate biophysical properties of the sRNAs is either by changing lipophilicity, and I will talk about one example for muscle, or by changing the size without profoundly enhancing lipophilicity, which is the chemical platform divalent sRNAs we prefer to use in CNS. Okay, so lipophilicity, how do you do it? There's tons of natural lipophilic conjugates, and we have done enormous amount of work and many others on chemical engineering of the space. And practically for systemic delivery, you can manipulate the PK/PD properties of the oligos by just manipulating the type of lipophilic conjugate it's attached to. So in this case, you can see that there is a lot of things which clearing through kidneys, there's a lot of things which are clearing through the liver. So the way one of the conjugates we love to use in the lab a lot, it's called doxaxonic acid. It's a 22 carbon tail, and it enables a very robust delivery to muscle, heart, lungs, skill, and others. And the reason behind it is that it binds to different classes of serum proteins, and systemic distribution of lipophilic acids is driven through this binding to the serum proteins, because if it doesn't bind, the sRNA, even fully chemically stabilized, it's a large small molecule. So if you inject non-conjugated sRNA IV, 95% of injected dose is going to be cleared through the first passage to kidney. So you're just making a very expensive concentrated solution of oligo in the urine. There is no availability. By this type of lipophilic conjugation, you can expand to vast majority of injection dose to stay in circulation and deliver to tissues. I love this slide because it's very visual, and the one of the targets which is popular in the lab right now, it's myostatin. It's an inhibitor of muscle growth, which is relevant to actually a wide range of clinical indications because it's enable growth of the muscle if you silence it, and this is how their ball with mutated myostatin look like. You see, it's my personal dream, maybe one day not to need to go to the gym ever again, because you can just make this, get this sub-Q injection. What is interesting that about this target that you can use it and monitor a lot, and it's a very easy phenotype to see. And in this particular experiment, we compare the durability of myostatin silencing in circulation, which you can measure without killing mice, where you can see a single sub-Q injection in mice gives you approximately 2 months of silencing at 80% silencing. And another experiment Hassan Fahih did, he actually was injecting it to every 2 weeks at relatively high dose for 6 months, essentially to be able to see the tox. And we didn't see the tox, but you can silence pretty much myostatin below level of detection. What is interesting over here is that independently whether you silence it for a short period of time or if you maintain silencing for long periods of time, you actually do get increase in lean mass and you do get increase in muscle strength. What was interesting about this study, and it's published in NAR, so please take a look if you want to listen details. It's really allow us to discover one very interesting fundamental feature of lipophilic assays, that their PK-PD relationship is not linear. What do I mean under that? So if you do a single dose injections at different doses, so you went from 5 to 40 mg per kg, so it's 8-fold increase in a dose. And then you look at the accumulation in the different tissues. You can see is that in muscle, 8-fold increase in a dose resulted in more than 2 orders of magnitude or up to 2 orders of magnitude increase in accumulation. Yeah, so 8-fold increase in dose, around 100x increase in accumulation. But this is true for muscle. This is not true for heart, because in the heart, the relationship is actually linear. There is maybe another way to describe the data, but what we can see is that in rodents, doses below 20 mg per kg do not deliver to muscle effectively. They do deliver to heart because you need to saturate the liver first. So what you see on the graph on the right, that at low doses, the fraction of injected dose, which accumulated in the muscles, is very, very low. It's like 1% or less. But if you go to 40 mg per kg, up to 25% of injected dose, it actually accumulates in muscle, giving you a very robust silencing. What is important is that this kind of nonlinear PK-PD relationship, it's very species dependent because it's dependent by metabolic rate of the liver. And one of the compounds developed in the lab a long time ago based on this type of delivery platform. And in this case, we were targeting soluble FLT1, which is a cause of preeclampsia. And in this particular case, the key is to silence the circulating form without affecting the membrane-bound form, which you can easily do with siRNAs. And Comanche Biopharma, and this is publicly presented data at OTS by Comanche, they actually look at this PK relationship in different species. And what they have seen is that it's dose normalized for lipophilic assays, the tissue exposure is order of magnitude higher than for the GalNAcs, which is interesting. It's really what drives this extrahepatic delivery, and it seems to be that the doses about 1 mg per kg, you actually start to see already liver saturated in a lot of extrahepatic delivery. So what we know right now about the platform, that it's chemically synthesized, so you can make this compound in 4 hours. There is no need for another conjugation, which make it very academic friendly. It's cheap. In rodents, you need to do high dose to get towards extrahepatic delivery, but in clinic, 1 to 3 mg per kg every 3 to 4 months expected to be therapeutics. So how do you apply this concept if you can get a chemical or delivery, systemically delivery to the muscle? And the story I'm going to tell you, it's actually the work collaboration between Julia Alperman, which is tenure track professor at UMass Chan, and Jay Shim. And I think to make a drug out of oligo in a context of academia, collaborative efforts is absolutely essentially, and usually it's a collaboration between people who understand the technology, and in this case, it's Julia's lab, and people who understand the disease and have animal models, which is Jishin. So they work on a very rare disease. It's called fibrodysplasia ossificans progressiva. And it's absolutely devastating disease because what's happening is that over time, with any injury, like you can pinch the muscle start to convert to bone. So the bone is developing inside the muscle, which of course results in loss of motility and kind of devastating quality of life. So biologically, this disease is 97% of the patients, the disease is caused by the single nucleotide mutation. It's this G to A mutation in ACVR1 gene. And ACVR is a signaling gene, which is responsible for activation of the bone growth. And as you can imagine, it's absolutely critical. You cannot delete it, you cannot silence it completely, because if you do, you will stop bone remodeling in the whole body, which of course is not compatible with life. So if you want to develop the treatment for this disease, you need to silence the mutant allele without affecting the wild type. And I think it's a very interesting story because in a lot of NO1 indications, it's actually critical to silence. There is a gain of function mutation, and it's sometimes gain of function mutation in a gene which otherwise an essential gene, and silencing just the mutant allele without silencing the wild type is critical for development of therapeutics. A while ago, we have done it, and this is not a confidential slide, it's published in Nature Communication, but we developed this strategy copying a lot of other people kind of using the same context, that if you want to develop a SNP selective compounds, what you can do, you essentially walk around the SNP. And what you can see when you're walking around the SNP, and in this case, in the lower the bars are better, and the black is on-target activity, and red is off-target activity, non-target activity, and essentially you want the black bar to be low and the red bar to be high. So you get some SNP selectivity, and you can see over here it's around 7x if you do the dose response. In vivo, this compound is completely worthless because this like 5 to 7-fold selectivity does not translate in vivo selectivity, because in vivo, of course, accumulation in different regions of the brain or muscle is different. Your durability of the effect and amount of compound inside the tissues at different points is very different. So while you compensate for it, you actually pick one of the leads where, and we have screened a lot, if you pick one of them which have some level of differentiation, and essentially you add the second SNP, second mutation, so in a context of wild type allele, you will have this one extra mutation, but in the context of mutant allele, you now will have two. And this actually results in a profound increase in discrimination. And this compound has 60x discrimination, which is already sufficient to see things in vivo. But if you need more, what you can do, you can block the stereochemistry next to the SNP site by including internal vinyl phosphonate. And that does not affect on-target activity, but it's minimize risk kind of wandering around the intended site, and in this case, it enhances selectivity more than 1000x. And this is just data in vivo in a Huntington disease model showing you that you can silence mutant without affecting wild type. Okay, how do we apply it to FAP? We did exactly the same thing, yeah. This is a SNP which we need to silence. So we did a primary walk, identify the leads which give you reasonable silencing, and then did a secondary walk to look at the best possible discrimination. And this is now validation of the lead compound, which has two mutations compared to wild type allele, and it's one mutation, but silent mutation compared to the mutant allele. And as you can see over here, you have a profound discrimination between mutant and wild type. And in patient-derived cells, originally you have mainly mutant being expressed, and after the treatment, you only have wild type expressed with effectively indetectable mutant amounts. What was interesting about the FAP as a disease, and as I mentioned, any injury results in this initiation of the mutant growth. And there is enormous amount of work going kind of in understanding why. And this is driven by local inflammation, which of course is driven by interleukin 1. So what we did, we screen a lot of, we confirm in vivo that indeed IL-1B is a major cytokine which has been activated. What is interesting for us that we published and developed a platform which is called this multivalent compounds, which can show that you can make one compound which will target in two different genes simultaneously. So what we did was screened a lot of compounds against IL-1 beta and identify very potent leads. And then the idea was that we essentially mix it together. So what you see here is a summary data, and this is non-targeting control. You see tons of the bone growth. ACVR, SNP selective compound by itself remove significant amount of the bone growth, IL-1 beta also, but the combination was most impressive. You essentially have non-detectable amount of the bone growth. So that is how the lead looks like right now. It's a bifunctional SI, which has lipophilic tail, and we were very Katie Gross, a former undergraduate student in the lab, and right now she's MD PhD student in Yale. She was able to get a hand on some discarded post-surgically discarded muscle tissue, wild type muscle tissue. And what you can see, validation of the leads, that it's absolutely silence IL-1 beta. And what was important for us to confirm in truly human tissue, this compound does not silence the wild type ACVR, so it's both effective and selective. So we are in the process of taking this lead and now advancing it towards formal clinical evaluation, and I will keep you updated on how things are. So let's go back to how you can manipulate biophysical properties. So we talk about attaching the lipid, but another approach which has a wider therapeutic index, because as soon as you attach the lipid, you're creating a little bit of a SOAP compound. Yeah, you have a negative charge and the lipophilicity. This is how SOAPs are being made. And of course, we engineer it in the way that this linker is cleavable, so to reduce the half-life of the SOAP within a cell. So within 24 hours, lipid is cleaved and metabolized, and you only have the SI in, but still, with local administration, therapeutic index is relatively limited. So another approach is just to increase the size. And what we have shown, and this is Nature Biotech paper in brain, showing that you get very wide distribution in the brain, you get a lot of retention in the lung, you have a lot of retention in the eye, and it's essentially a platform which allows you to uh, maintain a safe long-term silencing upon local administration. In the brain in particular, what was excited for us to see is that with non-human primate studies, you start to get this delivery to deep brain structures, which is essential for a lot of NO1 indications where it's not only cortex and spinal cord which are being affected. Okay, chemistry matters and chemistry innovation matter, and I will kind of talk for a couple of slides about this new chemical backbone called XNA, which was developed in Ken Yamada lab, who is also tenure-track professor at UMass Chan. And what is beautiful about this backbone is that it doesn't change general biophysical properties of their phosphodiester backbone. Yeah, it doesn't change lipophilicity, it doesn't change the charge, it just increase the distance between two different phosphates slightly. And what happens is that a lot of exonucleases and endonucleases are really sensing this distance between phosphate. And adding this XNA modification profoundly enhance the stability. When you enhance the stability, you increase area under the curve and accumulation in many different tissues. What was also very interesting for us to see is that if you apply just 2 extra carbons in the 3' end of the guide strand, you also get a much better potency at low doses in CNS. That is a paper on another compound targeting SOD1, which is also advancing towards clinic, where you add these 2 extra carbons and you actually further enhance a durability of the effect. And what you see over here on the graph is a comparison of impact and durability of the effect on the survival. This is antisense. This is approved drug, the only treatment for SOD1 ALS. It actually makes a significant difference in clinic, and it's first and so far the only one RNase H mechanism of antisense approved. But with divalent SI and with extra XNA, you get a profoundly longer duration of effect. Okay, which gets me to the last topic of today of how you actually advance exclusively in academia without for-profit sponsors a compound towards clinic. And as I mentioned, collaboration makes a huge difference, and the work I will talk about, and I think is a collaboration with a fantastic husband and wife team at Broad. It's Valab Medical Labs. This is Sonia Valab and Eric Minical, and they're amazing. They're great scientists. They're very passionate, and I think they're making a difference. The lab was started to develop the drug with Sonia because she is the one who is a carrier of mutation in prion disease, but it's I think they're making a huge difference for the field as a whole. So with a prion disease, what's happening? If you happen to have a mutation which prompts this change in conformation of the prion protein, or you get infected with a prion protein, this is like mad cow disease, it changes the conformation and then it aggregates. And when it aggregates, this is what causing lethality. And of course, the idea, which was circulating for quite a while, you should lower the prion. And if you lower the prion, which is non-essential gene in adults, you should prevent that. So We wanted to use siRNAs to do it. And how do you do it? You have to design it first. And I have no time to talk about it in detail, but our goal is to do it right, but then enable everybody to play with those tools. And over the years, we accumulated, I think, total 8,000 of intracellular efficacy of fully chemically modified SIs. And we work together with the Corkin lab in WPI with fantastic AI and advanced language processing computational science lab, and we developed the novel algorithms which gives you an ability to have a very high predictive power for efficacy of fully chemically modified SIs. Katie Monopoly, who graduated from the lab recently, and right now she is at Regeneron working with Bailey. They develop the web portal, and the address is very easy to remember. It's srna.ai. I didn't believe that this domain was available, but over here right now it's a better version of this portal, but you just put your gene name and it's generate you the lead candidates, which has a compatible CMC and pretty much all specificity, cross-reactivity, whatever you want. Okay, so let's go back to prion. So we predict a lot of compounds which are expected to be efficacious, and you need to screen a decent number. And what you see here is a screen at two different concentrations, and you start to identify your leads. You then perform the curves and measure the IC50 values in vitro. And I think what I would like to emphasize that the quality of assays matter because minor differences in efficacy can translate in a profound differences in clinic. Also, when you get the first hit, it's a good idea to walk around, and it's published and well understood strategy. And again, the differences are relatively minor, but if you compare this 2439 and 2440, they're next to each other. It's one base pair shift, but you get a little bit better potency with the 2439 versus 2440. And the last and not least, when you're moving in vivo, you have to move multiple compounds because the most potent compound in vitro, like for example, over here, it's 2768. It has 10 nanomolar EC50 values in passive uptake. It's actually not the most potent compound in vivo evaluation, and it gives you less overall prion silencing than 2439 lead. Okay, so chemical and structural optimization matter as well. And what Eric decided to do, he decided to move ahead with many different chemical architectures. And because Eric and Sonia worked with antisense quite a bit, and I mentioned this phosphatide, it's a beautiful modification. It gives you stability, but this is also the one which is dose-limiting tox. And he was absolutely married to the idea of trying to reduce phosphatide content as much as possible. And one of the chemical configuration which was advanced forward was a chemical configuration which only have two phosphatide modification in a row through the molecule. So there's no stretches of phosphatides. And of course, this was possible only in the context of this XNA chemistry because it gave you an extra protection. Another thing Eric and Sonia did because of the Bartel and Narakim work, talking about the perfect complementarity of the guide strand result in potential of recognition by other enzymes in the cells and cleavage, we optimize the 3' end. And what it seems to be is that if you do not have the matching to the target, the 3' end of the guide, you're gaining another like 15 to 25% efficacy in vivo. If you are using a tool compound for studying mechanism of disease, all of that is absolutely irrelevant, correct? But if you want to optimize the chemistry and chemical configuration to get the maximal silencing, those small things really makes a huge difference at the end. Right now, we are coming to a chemical configuration where essentially both the 5' end of the guide, which is always E-vinyl phosphonate U because it fits in the mid domain the best. We also optimizing the 3' end guide, 3' end of the guide to have a chemistry which fits the 3' and the past domain the best. And again, both of them are non-targeting, so you can come up with a uniform platform which helps with ensuring a very predictable PK/PD profile. Because again, 3' end of the oligo is where sequence matters a lot and where the sequence really does define the overall tox profile at the end. For example, if you have like a G at the 3' end of the guide strand, it's not guaranteed, but a lot of those oligos are very toxic. If you have fluoro G at the 3' end of the GANT, you will have the CMC problems during manufacturing. So unifying this region, it's critical, and right now we actually prefer to use UU, and in general making sure that it's very pyrimidine rich, because pyrimidines are, according to Ionis recent published data, and PNIT's tox is much more stable. So UNC reach avoid G's and H, and I will say G's should not be there at all because this is predominantly toxic. Okay, so at the end, Eric and Sonia have selected this lead, and it's a very interesting kind of learning experience because that was not my favorite. So they went ahead to this low PS XNA configuration. So again, as I mentioned, it doesn't have more than 2 PS in a row anywhere. And IC50 in vivo of this drug was around 1 microgram per gram, which is a couple of fold higher than what we used to see in CNS. And I think mostly it's due to the fact that it has lower PC content, so the stability is slightly compromised. At the end, Eric was right and I was wrong, because I was advising to go to the higher PS content, because by compromising maybe durability of the effect for the first clinical trial, Eric and Sonia gained something else. And what you see here is the results of dog and rat toxicology from the different doses tested. And what they were able to demonstrate that the 200 milligram, which is the highest dose tested, and just think about it, 200 milligram in dogs, it's correspond to approximately 3.3 grams in humans. That is very unconventional talk package design, and I think a lot of people advised Eric against going this way, because as a standard predominant opinion, at least in the pharma, is that you have to design your talks in the way so you would not see the talks, because if you would see the talks, you will need to communicate it to FDA. Actually, because Eric and Sonia labs are mission driven, their perception was, or response was, we would like to see the talks. We would like to know how far not to go. And that is why they went with this very aggressive dosing paradigm. And it's paid off, because 200 milligram dose in dogs was declared as NOAEL. which was shocking. And I think it's probably due to the fact that another decision was made to profoundly reduce the phosphatide content, which enables this very wide therapeutic index. There is a general saying by many philosophers that a lot of things you do today will define how you will live in the future. And this very aggressive and unconventional toxicology platform actually translated in a very interesting thing is that it's allowed very aggressive dosing paradigm in human. Okay, so how do you manufacture? The substance was manufactured in Hanjin, and they were fantastic partners. They were very supportive and understanding of NO1 costs, and I think they were extremely nice to us. I have to mention that the process optimization matters, and sometimes a lot of NO1 community think about the manufacturing is kind of benign thing, which doesn't matter. It does. And one of the interesting things we learn in the process that actually small changes in a manufacturing process, what type of reagents you use, may affect PS stereoisomers ratios. So by mass spec, and HPLC, it will look exactly the same, but it may affect functional performance. So the process optimization for the particular class of drugs you use matters a lot. Another interesting learning experience was the discovery, and I think this effect is much more pronounced with siRNA double-stranded oligonucleotides rather than with single-stranded, but in CNS, formulation matters. And in particular, what we discovered, not willingly, but if you are using isoflurane, like if you will use a long-term anesthesia, those effects are going to be masked. But if you're using isoflurane, so the mice are waking up right away, if you administering large concentration of the oligo, this is your intent. You want to have the silencing to last for long period of the time. What's happening is they're actually titrating calcium from the CSF, and calcium is a very critical signaling molecule. And when it does, you start to observe seizures phenotypes. Eventually, things do kind of mild out and balance out. But right now, we feel that it's absolutely critical to add divalent to supplementation to their formulation itself, and the formulation with which Eric and Sonia move forward has extra magnesium in a buffer. And if you're interested to learn more details of it, there is our paper in molecular therapy nucleic acids. And recently, Ionis also said, no, you can see this with antisense as well. And there is another paper now on essentially the same phenomena observed for antisense. So formulation matters. Read the papers by magnesium supplementation in the CSF. You can profoundly enhance therapeutic index even further. So with all of that, the drug was manufactured. This is actually it right now in several places, but here in Boston, in Broad, it's in the fridge. What Eric and Sonia did was also something completely unprecedented, because they say we invested 2 years of our lives to get this IND filed. We do not want other people to suffer the same, and they made the whole research IND publicly available. So if you go to this link, it has first full sRNA research IND public. It's 3,500 pages. So if you're interested, please go in. And the paper is right now just showed, I think last week it was published in NA. And as of today, there's several sites opened, and PRNP targeting siRNA safety mechanism of study, PRISM, is actively recruiting. So as of today, a lot of chemistry have been developed. It's all publicly available. Just pay attention. We would be happy to advise if people have questions, but we publish everything without anything being hidden. So please read. Manipulation by physical properties gives you ability to enable extrahepatic delivery. And things are in clinic in CNS, and hopefully with this data, we will see more and more compounds moving forward. And with that, I would like to acknowledge fantastic people I have known of working over the years, and many collaborations. And of course, I spend 50% of my time writing grants, and maybe NIH will eventually start back paying money, but we are highly appreciative of all the funding agencies. And thank you for your attention.
Thank you, Anastasia. What a wonderful overview. And it's amazing to see how the N-of-1 and ultra rare concept have really grown and expanded into all of these additional chemistries, really cutting edge chemistries with your leadership at UMass and RTI. We have a couple minutes for questions, and wanted to open up to the field to see. Please just raise your hand if you have a question that you would like to ask Anastasia. Or you can, if you prefer, type it in the chat, and then I will review and then call on you. Okay. Why don't we go ahead and start with the first hand up, Mathis?
Yeah, thank you, Anastasia. This was excellent as always. I'm still admire the exRNA modification I was just wondering, it does stabilize so much the siRNA structure. At how much does it come at the cost of increasing toxicity and immunicity? And you haven't shown any data on that. Could you update us on that?
So that is in process. We have seen, so the only possible potential toxicity can be coming from if it will get phosphorylated and converted to a triphosphate and then incorporated in mitochondrial DNA. We are in a process of doing those studies, but like preliminary, that is not biologically compatible for biologically compatible compound. It's like very inefficient substrates. This data will be generated. Other than that, and everything we have seen from a preliminary study, it's cleared as a whole oligo. So from our perspective, again, from it's not inducing adaptive immune response. And the reason why it's not inducing adaptive immune response, because what it does, it doesn't change the charge, correct? It doesn't change the sugar. It's only changing the spacing between the phosphates. And naturally, within a single-stranded oligo, the spacing between the phosphate, because it's flexible, it's not fixed. This is why, from antibody perspective, this is not a recognizable feature. I will say the XNA containing oligo will be in humans. It might be in humans now. I'm just not aware of it. So it's happening now. It's always very, it's scary because FDA, based on a very wide therapeutic index, allow Ericksonia to go ahead with a starting dose of 50 milligram, and 50 milligram is expected to be efficacious. But whenever you administer first time in human, it's scary, but we will learn more. And one of the things which Eric and Sonia did, which I, again, 100% supportive, but it's also scary, is for full transparency, meaning that clinical trial data will become publicly available in a real time. Meaning not necessarily the day of administration, but it's not going to be a typical pharma thing where you enroll the patients and then there is no data for several years if the drug get approved, and then there is no data never if you see something. So good or bad, we will learn very soon.
Thank you so much.
Katrina, there's a question from the audience about scaling conversions for dosing. Would you like to ask your question? Perhaps she may not be available. In case you're not available, oh,
Yes,
there
I
you
am.
are. Go ahead,
Thank
please.
you very much. Yeah, so you mentioned anesthesia. Thank you very much for this talk. You mentioned that with the DCA siRNAs, in mice, you need quite a high dose, at least 20 milligrams per kilogram.
For muscle, I will say 40. For heart, you can get away with much lower because it is less saturation. There's a lot of blood volume. Yeah.
Okay, but then you mentioned how it translates to human. Can you talk a bit more about that?
So the only piece of data we have so far is whatever Comanche has publicly presented, but they will be giving a very extended PK/PD talk on different species conversion of lipophilic ascites systemically at RTI Symposium end of June. So if you're interested more, please come and listen. I think the meeting is unfortunately full, but there is online version. And practically what they have observed that this liver metabolic rates are very different in different species, correct? And this is a well-known fact with the small molecules that rodents have a very high metabolic rates, non-human primates lower, and humans even lower. So far in the data presented publicly, even above 1 mg per kg, you start to see a lot of extrahepatic exposure in humans. You cannot, of course, do biopsies in humans, what Comanche has done, and Scott Johnson talked about it at, I think, at Cold Spring Harbor meeting or TS meetings, is that you can measure the area under the curve and estimate PK. So if when liver is absorbing oligos, your clearance is very fast, correct? When liver is saturated, the oligo is start to circulate in circulation for much longer periods of time, and this is what give you broad extrahepatic delivery. So what Comanche publicly has presented that the Cmax in humans is above 13 hours. So when you start to think about it, the oligo is like even at 1 mg per kg, yeah, it means that there is enormous liver is being saturated at very low doses, and then you start to see a lot of this prolonged circulation and a lot of extrahepatic exposure. So the phase 2 clinical trial is ongoing now. It's again very nerve-wracking. I do not know when. It's a pregnant woman, so they have to be very careful with recruiting and kind of dose acceleration. But hopefully, early next year, we will have the clinical results and efficacy. But so far, if you talk to them, they are saying that their perspective, based on all the PK/PD data in different studies, that the expected functional range for DCA class of compounds in humans is between 1 to 3 mg per kg.
And I have a second question. You mentioned about the DCA, like the linker being cleavable and DCA being freed from the siRNA, and you mentioned that after 24 hours, that's pretty much free. So is there
Yes.
any
Again,
research on this part?
that is published, no strings attached, no IP attached. What we incorporate is 2-deoxy-Ts. So it's naturally metabolizable, no new chemistry. So 2-deoxy-Ts without phosphatidated, it's metabolically stable in the circulation. You start to lose a little bit. We're probably in humans, because circulation is much longer, you're probably losing 10 to 15, maybe 20% of the drug during the circulation due to cleavage, but vast majority stay intact, but as soon as it get internalized inside the cells in endosomal lysosomal compartment, it's cleaved right away. So factually, if you look next day and purify, like get lyse the tissue and look at by mass spec what is going on, the sense strand conjugated to the lipid, you only can see traces. So everything is cleaved off. And then the SI itself will stay in cells for very long periods of time. That was intentional, because we know from gallnags that the oligo by itself, it's pretty safe. You can keep it in livers for a year, and there is essentially no observable effects. So idea was to mimic this phenomena while we're using lipophilicity to get the delivery, but then it's getting cleaved right away. I think you can optimize the space more, maybe not to lose this 50 to 20% during the distribution, because this is a waste, but we were, it's a first thing, and I was absolutely kind of paranoid about limiting the half-life of having a lot of mild soap in the lysosomes of endosomes of the cell.
Where do I find this publication about the cleavage and the
I think it's either NAR or molecular therapy. So I think the first author is Biskens. So if you do the search Biskens Quarava, it should pop up. And also
thank
we
you.
published for as Fleet, the original paper is Nature Biotech, and then there is a follow-up paper on chemical optimization and mTNA, where we talk about DCA phosphodyl DCA-amide, that's optimization of the lipid to reduce monocyte uptake. And there is a paper, I think Fahik et al. And NAR just published maybe December last year on further kind of chemical optimization in the context of XNA.
Thank you very much.
Fantastic. We are well over our allotted hour, but I wanted to thank Anastasia for joining us for an amazing talk. It's fantastic to see these targeted therapies, many of them first in class, preeclampsia, FOP, prion disease, and more, advancing all the way from structure all the way to the clinic here through the work of you and your colleagues. So congrats to you, Ken, your other colleagues, and all the trainees that contributed to that work.
Thank you. Thank you, Tim. We are committed to a community, and again, we publish everything. Like we are not there's no hidden secrets. And you're always welcome to ask questions.
It's a wonderful community that Anastasia is part of and is leading at UMass RTI. So I encourage others to take her up on that offer. I know I have. I will go ahead and formally close. Thank you all for attending. Please do continue the discussion on connections and reach out, where you'll be able to find the seminar recorded, and there'll be a chance to engage in conversations with additional questions on that site. And please also, as you exit, please remember to complete the survey, which will pop up as on your way out. With that, thanks again to our wonderful guest and all of you for your attention and your excellent questions.
Thank you. Bye-bye.